We should go on. We're running over. So the next speaker is Nathan LeBrasseur from Mayo Clinic in Rochester, and Nathan's going to talk about targeting senescent cells for healthy aging. Thanks very much, Nathan.
Thanks, Peter, and thanks so much to you and Gary and Mason for hosting this meeting. I have to admit, I'm more of a fan than a contributor. This is not the domain that I typically work in, and I'll do my best to make a comment about our interest in the epigenetic space or our fascination with this space. But my talk will largely be centered on this area of senescence and really an effort amongst many different institutions to come up with strategies to target these cells to improve healthy aging and longevity in line with the theme of this symposium. So I'll quickly disclose, we do have some intellectual property relevant to the work that I'll share licensed to commercial entities. And I should also disclose that I don't pretend to have any expertise in the epigenetic realm. So I want to be upfront about that. Quickly, a shout out to the team members that have been really major contributors to the work that I'll share today. Those individuals are pictured, and I'll try to call them out as I go through the slides. And then on the bottom left of the screen are other members of my lab that help support these initiatives. And as many of you know, many of us at Mayo Clinic have a strong interest in the space of senescence, including my collaborators, Joao Passos and Sandeep Khosla, who have been major contributors to the work that I'll share. And others that are listed, as well as Jim Kirkland and Tamara Chiconia, who I neglected to add to the screen. So I'm really excited about the overarching theme of this session of the meeting, and that's really this idea of intervening on aging to transform human health. And without question, this concept has radically transformed to science fiction to science now. And of course, many of us are kind of, I think, in awe of the headline grabbing news and scientific discoveries that have been dominating even the lay press. And that's because I think we've moved from the stage of better understanding the fundamental biology of aging. And of course, there are a number of discoveries to still make in this space and advance, but we're at this new era of asking the question of, can we intervene? And whether you're interested in fundamental markers of DNA damage that I've listed on the left, or the more recent insights into misinformation coming from that DNA because of epigenetic alterations, the opportunities for partial reprogramming, or more recently, this idea of stalled transcription, the notion that we can intervene to have an impact on human health as we know it is really quite exciting. And the notion, of course, is not to just make better blood pressure medications or better insulin sensitizers or bone building drugs, but the idea is if we can intervene on this fundamental biology to delay the onset of these diseases as a group. And of course, this is the concept that many people are now familiar with across many fields, not just aging, that we could extend human health span and really the impact on society would be compressing morbidity and extending quality of life. So this is a big excitement. And of course, we've heard a lot about epigenetic alterations and different strategies to maybe modify that landscape. But what I'll talk to you about today are some of the kind of trials and tribulations and the successes that we've had in targeting this other area of biology of cellular senescence. And I should emphasize that as much as we like to put these different forms of damage or misinformation or stalled replication into different buckets, clearly, when you impact one, you impact the others, both positively and negatively. And there probably is some degree of a unifying hypothesis that we could pursue here. And certainly, those interactions provide ripe grounds for interactions between our teams. So I'm not going to spend much time on talking about the fundamental biology of senescence. I think much like Dr. Sen said about epigenetics, it's diverse, it's complex. The phenotype and characteristics of one senescent cell may not replicate another senescent cell from a different tissue or origin, but we certainly agree on some core properties of senescence that I'll highlight throughout the talk today. Increased expression of some of these cyclin-dependent kinase inhibitors, alterations in DNA damage or telomere associated foci, increased expression of the senescent cell anti-apoptotic pathways. It's kind of fascinating to think that these cells are damaged to an extent that doesn't cause death, but they actually turn on pro-survival machinery to stick around and avoid apoptosis. And a number of other characteristics that I have highlighted in this diagram. And I'm really showing this to emphasize the point that over the years, since the kind of fundamental discovery is that targeting senescent cells can impact different parameters of health, there has been a laundry list of studies looking at how senescence influences the health of a number of different organ systems. And I would argue that those different studies in mouse models highly replicate that slide I showed prior that showed our concerns in human health about diseases across a number of different physiological systems. And it's really quite impressive to me that the idea that targeting this one cell type can have marked effects on osteoporosis and osteoarthritis, the manifestations of metabolic dysfunction or diabetes, fibrosis and dysfunction of the lungs, alterations in kidney health and function, plaque formation in our vasculature, the development of plaques and tangles in our brain, and the impact that those senescent cells not have just on driving damage, but really squelching the regenerative potential of different tissues. So it's a bit of this two-for approach of when we look at how these cells contribute to disease and damage. It's not the damage they're causing, but they're certainly creating a toxic environment where regeneration is severely compromised. And of course, more recently, in light of COVID, a strong interest in how senescent cells may be amplifying some of the cytotoxic storms that occur in the context of these different types of infections. And really the exciting part about this is we started early on with different genetic approaches to target P16 expressing cells, or more recently P21 expressing cells, but that quickly evolved into looking for different ways of pharmacologically targeting these different cell populations. And admittedly, we're very, very early in this space. I think we have a number of interesting proof of concept molecules, but we have yet to have really large engagement with pharma to start looking at large drug screening libraries where we can look at different strategies to more specifically and effectively target these cells in a safe fashion, and then go from there to hit to lead optimization and really have better drugs. And kind of as I walk through my story today, I want to emphasize this point about really the heterogeneity of senescent cells, and I'll use one tissue to kind of highlight that point, and how we may need to step back and think critically about whether one drug that targets senescent cells, either by killing them, which we refer to as senolytics, or changing perhaps their behavior, such as their secretome, which would be referred to as xenomorphics, we may need a very different cocktail for one constellation of age-related conditions and another. So just keep that in mind. So I've been very fortunate to help with some of these projects and lead some of the projects highlighted on the screen at Mayo Clinic and other institutions. But I have to say, for the past 10 years, I've been walking around with a little bit of a chip on my shoulder because I'm really a skeletal muscle physiologist. And I've been a little bit frustrated by the fact that there really hasn't been much published on whether or not senescence is even relevant to skeletal muscle, a tissue that's largely regarded as predominantly a postmitotic tissue. And other than a story from Pira Canoves showing that satellite cells may express a little bit of P16, that if you tinker with that, you can improve muscle regeneration, there really hasn't been to date a thorough or methodical examination of skeletal muscle and its susceptibility to this age-related form of damage. And importantly, I'll just highlight that as much as we think about muscle as being this postmitotic tissue of these huge myofibers with multiple nuclei, 50% of the nuclei in muscle are actually from these other mononuclear cell populations that may have more susceptibility to senescence. And we recently looked at the literature to kind of collect different information on what is known about senescence in skeletal muscle. And really, there's hints of senescence at a bulk level in muscle, but when you start to look at the individual contributing cell populations that may be prone to senesce, there's very little known. And this is just kind of a 30,000-foot view of the literature and the level of evidence that we have for senescence features in these cell populations. And one of the challenges that our field has faced is clearly the fact that there's not a standalone marker of a senescent cell, right? So we've tried to use this kind of comprehensive list of core properties of senescence that include factors such as what is inducing senescence, is it a form of DNA damage, metabolic stress, mitochondrial dysfunction, telomere related damage, or what it might be, as well as the cyclin-dependent kinase inhibitors, which have really dominated the field as kind of readouts of senescence. The presence or absence of a secretory phenotype, and then of course a number of different cell type markers, which I'll share with of different forms of damage and dysfunction within organelles that I'll share as I go through the talk. And clearly, I hope I don't have to make too hard of a sell of why muscle matters, but clearly it's an important organ for maintaining our health and independence into late life. There's just a discussion about metabolism, and muscle is, of course, the largest metabolic organ in our body that directs our resting metabolic rate, stores all of our glucose, and does a bunch of other important things. And of course, in this concept of frailty as we age, muscle is clearly a key organ for that. So that's why we've been so excited about it in our scientific career. So let me just start by telling you a story that was recently published and led by a postdoc in my lab, Zhou Zhang, and he really kind of used the mouse as a system to investigate senescence in skeletal muscle. And I'll just note that muscle aging is a relatively late event in mice. So we look at mice that are at least 24 months of age to study this phenomenon. And much like humans, mice gain weight as they get older. And despite the fact that they're gaining weight, they lose lean mass and muscle weight. And this is just kind of a global overview of what muscle aging looks like, or some people refer to it as sarcopenia. And when we look at a number of different muscles in the mouse, whether these kind of fast oxidative glycolytic muscles, such as the EDL that are important for force production and rapid movement, or slow oxidative muscles that are important for more endurance type activities, we can see features of senescence, such as high signals for P16 and P21 across different muscle groups, both in male and female mice. And on the right-hand side is just a poor man's heat map using qPCR to show some differential expression in the muscles of old mice compared to young mice, suggesting that there's a theme for senescence occurring in this tissue. And really, we partnered with João Passos' team to look at more kind of defining features or complementary features of senescence in skeletal muscle. And really, these are beautiful images to show that some of the markers that João really likes, such as telomere-associated foci, are highly enriched in the muscle of old animals at a bulk tissue level. We see alterations in centromere length and the loss of lamin B1 and HMGB1 from the nuclear envelope. So we see these kind of changes in core properties of senescence that have been used to define senescence in other tissues throughout mammals. And while this is all fine and good, we were really interested in, well, what is the source of these signals in a tissue with multiple different cell types? So to start, we use single cell sequencing, and admittedly, that's a somewhat of a restricted view of the landscape of muscle, because these large multinucleated myofibers aren't selected, right, for putting into droplets and sequencing. And just the kind of the processing of muscle does yield some myonuclei because some of the cells can rupture, but really the focus here is on the mononuclear cell populations. And those are kind of detailed in the middle panel with about 10 different cell types that we extract in the single cell sequencing approach. And on the bottom, we see some age-related changes that are consistent with what we know in the literature, or through different staining techniques where there's maybe a loss of capillary density within muscle, a shift in the satellite cell abundance, and an increase in pro-inflammatory macrophages. But what really stuck out to us were two things. One is that somewhat unexpectedly, it's these mesenchymal fibroadipogenic progenitor cells that demonstrate the highest abundance of a P16 signal, which is enriched in old animals compared to young animals. And the second element is that what we can more readily detect in aging skeletal muscle tissue in humans and in mice is a P21 signal. But none of the mononuclear cell populations were exhibiting any shift in P21 abundance in the mononuclear cells in the old mice. So we were quite interested in the fact that there seems to be this distinct P16 signal coming from these fibroadipogenic progenitor cells. And we can talk more about those in a bit. When we look at that cluster of FAP cells, we see that they divide into four unique clusters. And it's that third purple cluster on the screen that has this high P16 signal. And we look at this high P16 signal as accompanied by other features of senescence, such as increased enrichment, I should say, for different cytokines and chemokines and matrix remodeling proteins that we associated with the SAS. And on the right-hand side of the screen, when we look at what pathways are enriched in that cluster of P16 positive FAPs, there are many signals and cues consistent with a senescence program. And as I go through the talk and we use these different strategies for gene set enrichment, you'll keep seeing some features such as chemokine signaling pathways, cytokine receptor interactions, toll-like receptor signaling pathways emerging in senescent, what we think are senescent cell populations. And of course, when you enrich for those pathways and look at how they differ between that cluster 3 of P16 positive FAPs and the P16 negative FAPs, you can see it's highly enriched for chemokine signaling in that cell population that we think is senescent. So as much as we're really excited about the single cell seq data, we thought we should take an orthogonal approach to show that it's truly the FAPs that are emitting this signal that we're detecting in terms of P16. So we did a sorting techniques using MAX. And that middle panel shows how with that MAX technique, we could nicely isolate the CD31 and CD68 positive endothelial cells and immune cells, using PAC7 to isolate the satellite cells, using PDGFR alpha to isolate these unique FAPs. And you can see that we nicely separated these cell populations based on the first four graphs. But when you look at that P16 signal, again, it's the FAPs of the aged mice that really have this enriched P16 signal. A little bit of a bump in the endothelial immune cell populations, and a little bit of a bump in the satellite cells, but clearly it's these FAPs that are harboring most of this signal. And then again, working with Zhou's team and isolating these FAPs to look at other complementary markers of senescence, we once again see that these FAPs themselves are enriched in aged animals with gamma H2AX within the telomeres, an increased length of the centromeres, and then the nuclear loss of lamin B1 once again. So really consistent and somewhat comprehensive features of senescence in this cell population. So what's not in that story is kind of our deep dive or our interest in better understanding what these senescent FAPs may do. And we're talking about a pretty small percentage, if even an entire percentage of the cells within a tissue. And clearly, one of the most fascinating elements to senescence to me is the senescence-associated secretory phenotype. And there's this very clever tool that came out while we were doing this work called Cell Chat. And that allows you to use single cell RNA sequencing data to look at different communication patterns in the cells within your analyses. And what I'm showing you here is kind of what popped out to us, and that's this kind of cool autocrine and paracrine signaling that occurs between senescent FAPs and macrophages and non-senescent FAPs and macrophages, and the FAPs between themselves. And this is a busy slide, but let me just show you a couple of things here that I think are really cool and underappreciated in our field. One is that when we look at the outgoing signals from non-senescent FAPs, they're quite abundant and consistent with the cell type that we're studying, right? So we see high signals leaving healthy FAPs, such as fibroblast growth factor, BMP signals, IGF. Those are very abundant. But what's interesting is when we think about senescence, we often think that these cells take on this very robust secretory phenotype, which is true. But what we don't talk about is what they're no longer secreting. And I think this is an overlooked area of biology that is fascinating in better understanding how senescence is not only contributing to a toxic milieu, but these cells are no longer contributing key elements to maintain tissue homeostasis. So I want to point that out. At the same time, the senescent FAPs are acquiring signals that are not present in their healthy counterparts. So here I've highlighted tweak and SPP1, and I'll come back to a couple of these in the next slide. The incoming communication patterns highlight how these non-senescent FAPs, we think, are conducting a lot of autocrine signaling. So the incoming communication patterns that are outgoing from the non-senescent FAPs, they are receiving themselves. So there's some signaling to the macrophages. But what we see in a couple examples, such as SPP1 again, is a robust signal here, and the main recipient of that signal is in the macrophages. So we've been interested more and more in how senescents are communicating with macrophages and potentially causing some changes in tissue composition. And to that point, I'll just show you one example here of an experiment where, as we try to understand how these senescent cells are changing the health of skeletal muscle, these cells are known to differentiate into adipocytes. They're known to contribute to fibrosis. They play a critical role in muscle regeneration. They may be influencing muscle atrophy, but we asked a question about whether they're influencing kind of the inflammatory state of an aged tissue. So when we induce senescence in these cells, primary cells in vitro, we look at their secretome by measuring the content of these different proteins in their media. And this is kind of a select list that we've developed as biomarkers of senescence. And in the media of senescent cells, you can see really remarkable enrichment of these proteins across the board. Some of them are growth factors, some of those are cytokines and chemokines, and some of those are different matrix remodeling proteins. And you can see these are increased whether we induce senescence with etoposide, a DNA damaging agent, or with irradiation. And when we do a macrophage migration assay, we can see this milieu of these senescent FAPs is clearly driving an increase in macrophage migration compared to the control media. And when we look at a couple of the factors that are uniquely enriched in these senescent FAPs compared to these non-senescent FAPs, when we add an antibody to that media, we can abrogate the migration of these macrophages in this assay. And this isn't to suggest that antibodies to osteopontin and CCL2 are the only ones that are active. We haven't done an exhaustive search, but it's kind of interesting to see that what we learned through a bioinformatics analysis seems to play out in an actual in vitro assay. So now I'm going to start sweating because this is the only any type of epigenetic story that I have or chromatin story that I have for this talk. And I'm very grateful to Sarah Jackham, who's a PhD student in my lab who's been doing this work. She came to me with a very kind of out there idea, the notion that shift workers have accelerated aging. So maybe there's an interplay between senescence and circadian rhythms. And she's shown through a number of different assays. This is just one flavor on the far left of how there is this kind of increased amplitude and phase of senescent, or I'm sorry, circadian clock genes or regulators these are fibroblasts in this case. And there's this shift in amplitude and phase is really quite interesting. And we see this high level of BMAL, which is coded by ARNT1 here. And when we do a ChIP-seq assay with BMAL, we see differential binding. And we're just in the point now of looking at the interaction of this BMAL ChIP-seq, which clearly has different binding motifs in a senescent cell versus a non-senescent cell. And as a pioneer transcription factor, it's interesting then to integrate that with the RNA sequencing data from those cells. And we're starting to get some insights into potentially how BMAL may interplay with a senescent cell and regulate gene expression. And we think this is maybe a key player in regulating the SASP. And on the right-hand side of the screen is somewhat unrelated, but we actually have this data in BMAL knockout cells as well. And you can just see clearly there are clusters of genes that are very differentially regulated between a senescent cell that has BMAL intact and a senescent cell that does not. And this will help feed back into understanding how this interesting biology is interacting. And I'll be reaching out to many of you to help us walk through these analyses and do complementary studies. So I've given you kind of a quick pitch on this interesting story about P16 positive fibroadipogenic progenitor cells. But where is this P21 signal coming from? And I've shown you that at a tissue level, we see high P21. But when we look at the mononuclear cells, either at the single cell RNA sequencing data or through the max sorting and look at the mononuclear cell populations, there's no evidence of an increased P21 signal coming from those cells. In addition, if we do, just look at a Western blot for P21 in whole skeletal muscle, we see high enrichment. And then if we do RNA-ish staining, we see more fibers in the aged animals stained positively for P21. And in particular, we see many more fibers with several punctae of P21 in the aged animal. So with these kind of preliminary data, my postdoc Zhou Zhang then went through the effort of teasing out 180 mature myofibers from an old muscle and 180 mature fibers from young muscles. So multiple animals contributed to this cell population. And what you can see on the bottom right here, that the majority of myofibers in an old mouse have the same amount of P21 as you see in a young mouse. This is a pretty abundant transcript. But there is a subset of these fibers that have incredibly high P21 levels. This is about somewhere in the 5 to 8 percentile range of all the fibers that we collected. When we look at the staining, it's kind of a similar number that we see having many punctae for the P21 signal in a tissue cross-section. So we thought this was quite interesting. So what Zhou did then is once he measured P21 using qPCR on these fibers, he then submitted young fibers, old P21 low fibers that had the same P21 abundance as the young fibers, and old P21 high fibers for sequencing. And what you can see here is really a dramatic difference in the transcriptional profile of the old P21 high fibers compared to either the old P21 low fibers or the young fibers. Meanwhile, the young fibers and the old P21 low fibers, even though they're only differentiated based on this one factor, are really quite similar, which was really fascinating to us. When we look at the enrichment and the different pathways in those old P21 high fibers, again, a lot of the same factors that we saw in the FAPs emerge, such as increased cytokine receptor interaction pathways, JAK-STAT signaling, of course, P53 signaling, consistent with the isolation of using P21 to isolate these fibers. And again, signals to us that despite the fact these are postmitotic cells, have many features or characteristics of senescence. And some of the genes contributing to these pathways are highlighted on the right-hand side of the screen in the heat map. So we're really quite interested in whether or not to understand the significance of P21. So we asked kind of a real simple question of whether or not P21 is sufficient to drive features of skeletal muscle aging. And we were fortunate to do a proof of concept experiment because Jan van Deursen had generated a mouse that had P21 overexpression, also with the reporter tomato built in. And you can see across all tissues, high tomato expression in both the control animals and the animals with both tomato and P21 overexpression. And throughout organ systems, you see effective induction of P21. And interestingly, this story really hasn't been told yet, but these animals have a lifespan of 3 or 4 months. They have features of aging such as kyphosis, graying of the fur, cataracts, and some other features that really have not yet been carefully characterized. They're somewhat of a difficult line to work with. But interestingly, when we looked at the muscle of these animals, and again, I'm the first to admit that this is not a conditional model, it's not tissue specific, but even so, the muscle here, again, has many of the pathways upregulated that I mentioned prior. And again, suggesting that this kind of transcriptional profile of a mouse with high P21 in the muscle, it nicely reflects what we're observing in a chronologically aged mouse, makes this an interesting model to look at. And what you can see here is a number of different markers of senescence, including an increase in gamma H2AX, are markers of DNA damage within the myonuclei of the mice overexpressing P21 compared to their controls. And in addition to that, we see other features such as mitochondrial dysfunction, which Zhou often reminds me is an overlooked feature of senescence. But here we see marked reductions in mitochondrial capacity and function in the isolated fibers of the P21 overexpressing mice. And then when we look at other features within the animal, we see a lot of the things that we saw in what I showed you in the first slide in terms of what does muscle aging look like. And even though we see reduction in body weight in these animals, this is largely attributed to the loss in lean mass. And we look at the weights of the different muscles. We see that there's a reduction in muscle cross-sectional area when we measure the diameter of these different fibers within muscle. And there's a loss of force by these muscles as well, no change in fiber distribution. And of course, other features of muscle aging include a loss of function. So we see reductions in maximal exercise capacity on a treadmill, and we see a reduction in grip strength. And I didn't have a lot of time to go into it today, but we've been quite interested in our human studies at looking at different biomarkers of senescence. And we think components of this ASP are really quite helpful in doing that. And we take a lot of flack for not being able to demonstrate convincingly in humans that circulating factors that we think are linked to the SASP are only from senescent cells. But here's an example where many of the factors that we're quite interested in, like GDF15 and some of the matrix remodeling proteins, TNFR1, we see high levels of these circulating proteins in these animals with high P21 overexpression, providing some level of support that what we're measuring in humans is in fact linked to a senescence program. So we've done quite a bit of work in this biomarker space, and this is somewhat of an awkward shift into just talking about some of our human studies where we've looked at some of these different biomarkers in the circulation of humans, factors produced by senescent cells. And we've linked these different biomarkers to chronological aging, consistent with the premise that senescent cell burden increases with chronological age. And we've linked them to different clinical manifestations of advanced biological age. So we've heard a little bit about frailty today, as well as different adverse outcomes after surgery. So when we look at individuals who have undergone surgery for heart disease or for cancer, we can use these biomarkers to predict those who are going to have surgical complications or rehospitalization, different important clinical outcomes linked to advanced biological age in humans. We recently published how well these biomarkers do at predicting physical function in older adults. This is a study of about 1,300 individuals in the late life intervention for independent study based out of Florida. We have a story and review looking at how these biomarkers link to pulmonary health and function, a follow-up study to our work in mice showing how senescence can drive pulmonary fibrosis, and clearance of senescent cells can improve pulmonary function, and some follow-up studies looking at a number of different health outcomes in humans. And I'll come back to that in a minute, but I'll just kind of share with you, as we think about strategies to improve the health of organs through senotherapeutic interventions, many of us reach for the shelf for Dasatinib and Quercetin, because brilliantly, Jim Kirkland's group showed how this can disable the pro-survival networks in many different cell populations. Unfortunately, we don't see D and Q to be particularly effective in muscle itself. And I say that carefully because we see no shift when we treat older mice with Dasatinib and Quercetin in the P16 or the P21 signal. But even so, we think that the systemic effects on other organ systems of D and Q ultimately translate to improvements in the transcriptional profile of muscle. So this is kind of interesting, and there's been some really cool work done where you can transplant senescent cells into an animal, and even though they don't take residence in particular tissues, they can influence the health of other organs through their secretory phenotype, once again emphasizing the importance of the SASP. So we used DQ in this study somewhat to appease reviewers. We weren't really crazy about the experiment. We saw a nice shift in the transcriptional profile within muscle, and interestingly, things that we saw upregulated in old animals compared to young were largely reversed in the old animals treated with DQ, so that was kind of cool. And we saw that in animals treated with DQ, we saw a relative enrichment of the P53 signaling pathway in the vehicle-treated animals versus the DQ-treated animals, where we really saw no differences between meaningful effect in the old versus the young. So I'll just kind of share that this is another look at just some of the other analyses that we did in this study, and really kind of interesting trends, changes in central nucleated fibers, and a modest improvement in grip strength, but we think these are probably more systemic effects than specific effects on muscle itself. And I'll just share with you that we've been effectively or actively studying or looking for different drugs in partnership with Paul Robbins and Doohong Zhou that may more specifically target the cells in muscle that we'd like to get rid of, in particular the FAPs. And we're a little bit away from looking for drugs for the fibers, but when we've screened drugs to target these P16 positive FAPs, we've had a couple of very interesting hits. Navitoclax, which is a BCL family member inhibitor, actually shows pretty good efficacy, but its safety profile is pretty atrocious, so we haven't been excited about that. But here I'm just showing you some data from this other drug. We use an IncuCyte system to screen these drugs, and we can look at a dying signal that's emitted from these cells over the course of 48 hours. And what you can see is when we screen these drugs, we can see a dose response with this one candidate, 753B, where with increasing concentrations, we see increased death in the senescent cell population, but in the control healthy cells, we don't really see meaningful effects, which is important. And that's quantified here on the bottom right. And this is just preliminary data, but we've done an early trial in partnership with Doohong Zhou when he was at Florida. Now he's at UT San Antonio. But we really see impressive effects on the molecular phenotype of muscle in aged mice. This is a 6-month intervention where strength was assessed every 2 months in these animals. But you can see a pretty marked reduction in P16 and P21, consistent with a reduction in the cell populations that we're most interested in, and some other features that I won't comment on today that we think impact muscle health and function. Again, we see this interesting reduction in central nucleated fibers. This is maybe less about degeneration and maybe more about kind of innervation of muscle in the context of aging. And we see beneficial effects on grip strength throughout the course of the study. So it's really an exciting time in the field as we go from kind of bench to bedside, or kind of the Mayo lingo that we use as knowledge to delivery. And there's certainly no shortage of review articles now talking about kind of changes or advances in this area of Senotherapeutic drugs, I would say, not necessarily Senolytic. It appears that suppressing the SASP can be just as effective, or can be more effective in some cases, for manifesting improvements in health. But I promised to Peter that I wouldn't neglect probably our favorite intervention to promote healthy aging, certainly the most evidence-based intervention that we have. And that's exercise, where we are kind of exercise evangelists. And even before our understanding of senescence, really its ability to counter many of the different forms of age-related cellular and molecular damage. And there's some evidence, and some of it might be controversial, about exercise being able to prevent DNA damage or promote DNA repair, clearly restoring hormesis to mitochondrial health and function, stimulating autophagy and countering inflammation. There's strong evidence there. And then we think about the accumulation of senescent cells with advancing age. We think a lot of that has to do with this unhealthy cellular milieu, where immune function is compromised for identifying and deleting those senescent cells from a tissue. And we think exercise may play a role in promoting the clearance of these cells, and ultimately having an impact on how many senescent cells we accumulate throughout the life course. and in turn preventing the onset of a host of age-related conditions that I've referenced throughout the talk. And I'll just show you a very simple example. It was almost a study of convenience that we were fortunate to work on. We have an active older adults program at our workplace, and this was a study of 30 individuals that were 60 years of age or older, relatively healthy, I would say, because they were participating in an active exercise program. So we assessed them before they started the program and afterwards. And in response to this program, not surprisingly, we saw improvements in strength, reductions in the time it took to do repeated chair stands, doing standard clinical measures of function, such as the timed up and go test. We see improvements there in these individuals. And importantly, of course, patient-reported outcomes of fatigue and well-being and quality of life also improved in response to this intervention. And we took advantage of Ned Sharpless's assay, where he isolates CD3 positive T-cells from the circulation as a biomarker of senescence. These cells are enriched for P16, so they're a little bit better in terms of signal to noise ratio for assessing senescence, signals, if you will. And what you can see here is that we saw a pretty consistent reduction in many of the markers of not only just P16 signals and P21 signals, but activation of the cGAS-STING signaling pathway in these individuals just 12 weeks after they participated in the exercise program. And of course, I can't claim that we reprogrammed the cells, but that's one possibility, or maybe there's kind of rejuvenation of these cells, and these are a different cell new cells that we're measuring here in these exercised individuals. But I think it's a cool proof of concept. And I'll kind of add one more example of a lifestyle intervention. We've just finished the analysis and we're putting together the story on calorie. So Zaira Aversa, postdoc in my lab, has been hard at work on this study, where calorie is a very fascinating study. It's the only kind of 2-year longitudinal study in humans of caloric restriction. About 200 participants randomized 2-to-1, 2 to caloric restriction for every one person assigned to an ad libitum diet. The goal was 25% reduction over the course of 2 years. The average is about 12%. So if you think about it, it's not dramatic, right? But it's probably a meaningful amount of reduction. Clearly conferred or conveyed many health benefits and cardiovascular risk and metabolic improvements. And we've looked at about 30 different biomarkers of senescence, and we see about half of them are positively affected, even in these younger, healthy individuals, in terms of their circulating abundance of different biomarkers that we're interested in. And these are a little bit complicated graphs because they're measures in change, not necessarily abundance, but showing a significant effect of caloric restriction. On the right-hand side, we took advantage of some data from Deep Dixit, where he had a nice paper doing RNA sequencing on the adipose tissue of individuals who just participated in the caloric restriction arm. And you can see when we use this kind of gene set that we recently published for senescence called Sen-Mayo, we see relative enrichment at baseline compared to 12 months and baseline compared to 24 months in individuals who underwent caloric restriction, suggesting that some of these different features of the molecular signature of senescence are benefiting in the adipose tissue of these relatively young middle-aged individuals. So a bit of a smorgasbord today in terms of my thoughts on Senolytics as a strategy to improve healthy aging and longevity. But what I've tried to convey to you is senescence is indeed a hallmark of muscle aging. Importantly, we see very distinct signatures of senescence in 2 very different cell populations in muscle. And there's just a lot of questions that we have surrounding that of why P16 but not P21 in FAPs, and why P21 but not P16 in fibers, and what does this mean, and how is that regulated? We kind of demonstrated in the paper that a proof of concept senolytic cocktail of Dasatinib and Quercetin had probably meaningful or important effects on muscle, but what we're really excited about is more targeted therapies that exhibit superiority, at least in preclinical testing. I didn't show you the data, but we had another kind of figure in the story about we did biopsies on 30 young and 30 older individuals and showed that many of those same enriched pathways that we showed you in the myofibers and the FAPs are evident in the bulk tissue of older humans, such as cytokine receptor signaling pathways that are consistent with kind of the senescence and pro-inflammatory state. And then importantly, as much as we're all kind of in the search of these new drugs to target these cells, without question, we have a very scalable and readily implementable number of solutions that are lifestyle factors that we think clearly have an influence on senescent cell burden and SASP. So as we move forward, we're working hard on understanding the mechanistic understanding of senescence and muscle aging, as well as in other systems, and very interested in how these senescent cells within a given tissue are influencing the health of the surrounding cell populations. And of course, through a number of different efforts and collaborations, we're trying to advance the current battery of drugs that we have available to target these cells. So I'll finish by thanking you for your attention. It's great to be here. I've really learned a lot and enjoyed the discussions, and I'd be happy to take any questions.
Yeah, wonderful talk, Nathan. That's really, really beautiful stuff. So we are running quite a bit behind, but we'll get some questions in. There's question from Ramin Sadri in the chat, which he raises, the very interesting point of reactive oxygen species, which I think is very interesting when we're thinking about senescence in the muscle, because obviously, on the one hand, ROS is thought to promote senescence, may also has some beneficial functions, and ROS is also produced during exercise, which is something that you've shown is beneficial. So what do you think is the role and the interplay of ROS in senescence and ROS produced by exercise in the muscle?
Yeah, I think you already hinted at it, Peter, and it's really a story of hormesis. And we're all a little bit guilty. It's human nature to think, well, if too much of something is bad for you, I don't want any of it. And it's been clearly shown that if you get rid of oxidative stress with exercise, you lose its beneficial effects on other organs. So it's clearly this story of hormesis. I think part of this P21 overexpression story is clearly driving some mitochondrial dysfunction and oxidative stress that's inducing the spread of senescence and more DNA damage. So it's keeping it in check. And I think, yes, exercise drives reactive oxygen species, but it also markedly upregulates the antioxidants. So important to have those in combination. Okay.
And I think there is just to note, particularly for this audience, there's effect. I think there's effects of ROS on the epigenome as well. So I to what extent ROS can be detrimental versus hormetic on the epigenome is something interesting to think about. Vera has her hand up. Do you want to go ahead, Vera?
Yes, great talk, Nathan. I'm sorry if I missed it, but maybe you said that. What do you think? What is the driver of this senescence? Does it have anything to do with telomeres, or is it through ROS and stress?
Yeah, I mean, the most consistent signals that we see, and again, this is just from kind of transcriptional profiling, it seems like DNA damage is the consistent theme. I would really like to think of more sophisticated ways to measure that. We're a little bit guilty of doing gamma H2AX, and we love partnering with Zhou and doing the telomere-associated foci, but I think that's really a fundamental question that we definitely need help on how to tackle that.
Thanks.
Thank you. Yeah.
And Jessica Prue has asked, do you see sex differences in of the effects of exercise?
Oh, the effects of exercise. That's a good question. I thought you were going to ask about senescent cell burden, and I'll tell you that every experiment we change our mind of what sex is more prone to senescence. It just seems to shift a lot. And now with some of our biomarkers, we're seeing some sex differences. We don't know how meaningful they are. And then with exercise, I don't think so. I think we've seen pretty universal responses. Interestingly, this is a total, not answering your question, but for exercise studies, we get better participation amongst older women than older men. So that also influences our interpretation of the data.
Okay. I'll just take the last question before we move on. I mean, something that you noted, which was, I thought, really interesting, was the loss of expression of some factors such as FGF in I'm not sure whether it was I think it was the senescent FAPs. So what do you, I mean, we've all been for many years, we're all very focused upon the SASP, but to what extent, how important do you think, particularly when we start thinking more about senescence in a cell and tissue specific context, not just fibroblasts in culture, how important is going to be the loss of expression and loss of identity of specific cell types when they become senescent?
Yeah, I think this is a real important feature for tissue homeostasis. And I think it's going to be very obviously cell type specific, but even, and I probably shouldn't say this, because I think it's such a cool story, but even when we look at these P21 high myofibers, some of the myokines or exerkines that you hear about, we see lost in those cells. And that's probably important for the local tissue microenvironment. But then we also know that through things like exercise, right? How is exercise influencing brain health? And there are some factors that are known to be released from muscle that mediate those effects. And it's possible that in the context of senescence, you're losing that. So I think it's a really interesting area. I'm just trying to think of an acronym to describe it. It's like the the loss of a SASP. I don't know, LASP.
Yeah. Well, I guess SenNet will hopefully shed a lot of light on this in tissue specific contexts, loss of identity associated with cell senescence. It'd be interesting.